Hey there, ladies and gentlemen, boys and girls. Today I want to talk a little bit about, kind of give a little bit of the story. Talk about the historical record of how we found the structure of DNA. All right. I want to kind of go through kind of the narrative of what happened, and I'm going to go. Obviously, this what happened here involved dozens and dozens of scientists over decades. I'm going to kind of trim down into a little short story here. But because I want to really emphasize a little bit about this process, that major scientific discoveries like the structure of DNA, it takes more than one just person sitting in the lab saying, I found it. All right. It's normally a lot of people having a lot of ideas and each kind of contributing their ideas to kind of a pool of knowledge that then one group or one person then can kind of take advantage of, put it all together, and then they kind of are giving credit for it. And now when I say people put their pools or resources together, understand, in the scientific community, it is a competitive field to try and be first. It is quite cutthroat, more than I would say any kind of sports rivalry. All right, these are people who put their entire lives in their intelligence and their work, and literally spend decades of their own lives to answering a single problem or a single question. And so when I say they're sharing work, they're more or less just trying to take advantage of other people's work and put it together. And no story is more true than that, than this one right here. All right, so the whole goal is to find the race for the structure of DNA. All right, and this whole race, and it literally was a race because the scientific community was on a race here, or was trying to find it first, started with kind of the firing gun to get it started was with the work by Alfred Hershey and Martha Chase. So what I have here is, this is kind of our angle, and I'm going to talk about various scientists and groups who kind of all contributed to this idea, ending with the group that gets the final credit. So just kind of an idea where I'm going here. All right, so we're going to start with Alfred Hershey. And Martha Chase. All right, I'm going to give a few dates just to kind of give you some context. So we're about the 1920s right here. All right. They were two scientists, and what they were trying to actually prove was what protein held the genetic information that we passed down from in one generation to the next. Right. So we knew we had traits like brown hair and green eyes. All right, that we could pass down to our children. There had to be some sort of genetic molecule that did this, some sort of molecule that passed it down to the next offspring, right? And it was, for the longest time, it was thought to be a protein because proteins do so much, all right? But through their work, kind of eliminating a lot of the macromolecules possibly could be, actually came upon the molecule that it is, all right? So while working with viruses and mice, they actually came across the idea that it wasn't proteins that had the genetic molecule, it was a nucleic acid. More specifically, this one found in cells called deoxyribonucleic acid. All right. So they discovered that DNA is the genetic molecule not proteins. All right, and with this was sparked a huge start to the race here. All right, so we have this first one here kind of going to this. They're the ones who kind of kicked this race off, because once this started, it's like, okay, we know this nucleic acid is the genetic molecule, is carries that genetic information, but what does it look like? And immediately it was kind of sent out, and everyone's like, we got to be the first ones. And universities are hiring top recruits and out of colleges and universities to be on their teams to try and be the first one to make this groundbreaking discovery of the molecule that holds all the information that makes us us. What does it look like? All right? And it had to meet a couple criteria here. All right. And the first one we're going to talk about, the first person to kind of do this, wasn't really looking for the actual structure, but a lot of his work made a big impact on this. He was kind of working on it. His field was a little bit different. His name was Edwin Chargoff. Oops. All right, Edwin Chargoff. And he was a biochemist, right? He did a lot of labs, and he was trying to figure out the composition of it. So we have this DNA that we can isolate from cells. We can't look at it and see it. DNA is too skinny, too small to be able to see it, right? So we can do some chemical experiments, and we're trying to find out some composition of it, right? What is it made up of? We know nucleic acids, or nucleotides, but what nucleotides? There's a lot of them, different kinds, right? And after a lot of his work, he came up with something called called Chargoff's rule. All right, which says that basically that DNA is made up of 4 nucleic acids: adenine, thymine, guanine, and cytosine. All right, 4 nucleic acids, but there's something very specific about that, how that's oriented here. Chargoff's rule states that the percentage of adenine seems to be always equal to the percentage of thymine, like they are a pair together, and the percentage of guanine is equal to the percentage of cytosine. All right? And so what that means is, for example, if I have a sample that is, 15%, I have an example here that is 15% thymine, that must mean that DNA sample also is 15% adenine, but also must be 15% adenine, which also means that that's 30%, right? That means 70% of the molecule is made up of guanines and cytosines, but they are equal to each other, right? So this would be 15, 15, 30. That means this is 70, which means this must be 35% guanine and 35% cytosine to make 100% of the molecule. All right. So that's an example there of Chargoff's rule. If he knew one of them, he could tell the other ones. Because he knew adenine and thymine were a pair. He knew guanine and cytosine were very nice together as well. All right, or equal amounts together. All right, this was happening because it was about the late 1920s. This is happening really early in the 1930s. Just giving some context. All right, we're in the 1930s here. All right, this is kind of rolling around. Some major people are putting a lot of work in. Oh, let's put Chargoff's work into here. Into the pool of knowledge. All right. Next one here is the first big player to really step into the game here. All right. This is a man by the name of Linus Pauling. All right. Linus Pauling was a big man on campus. He was considered one of the best chemists known in the world at the time. He had quite the reputation. He was also known to be quite arrogant. He was really smart and really good at his job, and he knew it. And so a lot of people thought he would be the front runner because he had done a lot of great work already. All right. And through his chemistry, a lot of his work, he proposed the first model. All right. He made the first model of what it looked like of DNA. All right. He proposed the first model, and what he proposed is a triple helix. All right. Think of a ladder, right? It has two sides, and then twisted. That's a helix, right? Now, instead of having a ladder with two sides, think of it having three sides, almost like a triangle, and then a twisting. All right. That was what he proposed, was a triple helix to the shape, and a lot of people think, or he kind of was like, solved, figure it out, right? But after, like all science, it needs to be peer-reviewed. After it was looked at through the scientific community, as he kind of presents it, here's the answer. A lot of people looked at it and realized that's not right. And that can't be right. The chemistry doesn't make sense, and it was rejected. He was pretty embarrassed by this and kind of disappeared afterwards for a little while. But one thing he did say, all right, wasn't a triple helix, but he was the first one to bring up the idea of a helix. Think of us like a spiral staircase, a twisting staircase. He was the first one to propose that. And that, even though it wasn't the right answer, his right answer, that part stuck. He decided that the race wasn't, he's like, well, this isn't worth my time anymore. He was kind of embarrassed a little bit by being wrong, and he jumped out of the race. All right? And this was about the 1940s. So we're already talking about a race that's taking or at least a couple decades. All right. Now these last two, I'm actually going to write them at the same time because they were working at the same time. Although one group got a little bit of a head start, they were working at the same time. This first one here is Rosalind Franklin and Maurice Wilkins. And Maurice Wilkins. And then kind of working at the same time, we have James Watson and Francis Crick. All right. So let's talk about Rosalind Franklin and Maurice Wilkins, first of all. They were a team that worked together, but they did not like each other. All right? Rosalind Franklin now remember, this is also a time period where women in science women weren't given a fair shake. Most of them were looked down upon. In fact, actually, Maurice Wilkins has already started working on this, and Rosalind Franklin was brought in while he was on vacation. And when he returned, he actually thought that she was first his secretary, and then second of all, it was just there to work for him as an employee. Later to find out that, of the two, she was actually probably more the accomplished. And that kind of put a chip on his shoulder. He did not like her very much because she was pretty well respected. From those who actually took her seriously. Again, being a woman, some people didn't take her very seriously. And so they might have not spoken highly of her. But those who actually took the time and knew who she was as a scientist knew that she was brilliant. And one thing she was really good at, and she had learned by using years of research, working for the British government, the Allies during World War II, was studying coal and how to more efficiently use coal energy. She learned a process called X-ray crystallography. All right, and she was brought on the team because of her expertise in this. Which is basically you put a molecule inside of a crystal and you shoot X-rays through it, and what it does is almost kind of makes think of like X-rays. So you shoot X-rays through you, and then you get a kind of a picture of your bone structure. It kind of does the same thing for molecules, and then from that kind of that X-ray picture, you can kind of take some measurements, and you can figure out certain things about that molecule. And her job was to use her expertise in X-ray crystallography on DNA, right? And she was very structured to do this, all right? She spent a lot of time doing this. And actually, she gave up kind of in the middle of her time. She gave kind of a lecture, a public lecture to the scientific community in the middle of her progress, and just kind of talked about her progress and how things are going. She didn't care about the race. She just cared about the science. And so she shared it out. And two people attending that were James Watson and Francis Crick, right? James Watson and Francis Crick didn't do any scientific experiments. They were a biologist and a chemist and a chemist and a physicist. They were model builders. They literally built models and tried to kind of see how these molecules, they build like literally out of tin, they'd like almost sheet metal. They cut out these giant, what they thought were adenine, thymines, guanine, cytosines, and try and fit them together. And they literally just built models instead of doing quantitative scientific experiments like Chargoff or Pauling or Franklin and Wilkins. They just kind of did that. And they watched Franklin's lecture and were kind of like, oh my gosh, she's definitely on the right page. Like, she's got this right. And they actually proposed their first model. All right, they built models. That's all they did. And they proposed their first model, and Rosalyn Franklin attended their conference about the potential discovery of it, and quickly told him, like, your chemistry's off. You're not taking the time to do the measurements to find out. They actually had it, instead of kind of like a ladders going up the side and then the parts on the inside, which is what they had kind of reversed. And so they had to kind of go back. And eventually Rosalind Franklin, with her work, she proposed the double helix shape. Oops. That iconic the twisting staircase, the twisting ladder shape. She was the first one to propose it after doing lots of research. But she proposed it, but she was like, I need to do the data. I need to triple check my math. Maurice Wilkins, feeling a little dejected by kind of being pushed to the side because of Rosalind Franklin's great work. There's a lot of questions about his motivations. Basically stole her data and gave it to Watson and Crick. And Watson and Crick, with what she had done so far, even 60% of her data completed, was enough for them to make a new model. And they made a new model, and they immediately published it as the correct answer. And after it was peer-reviewed by the scientific community, checked the chemistry, so on and so forth, they had guessed right. They had never done one scientific experiment, and the last thing that they needed was Rosalind Franklin's data. Was taken from them. And they proposed the model, the correct model that is today. All right. They published the modern model. Oops. All right, they did so by publishing it in 1953. All right, after the work of Rosalind Franklin or Wilkins and Franklin. All right, published in a one-page paper, one page. Something for this big, what you'd think would take longer, but they were trying to get it out there to win this race. They published it in Nature magazine in 1953 in an article called "The Structure of DNA." They were given credit for it. They had won the race. They had won the race, and they are given credit for discovering the structure of DNA. Although they never actually did a scientific experiment. Now, I don't want to take it away from it. They still had to take a certain amount of intelligence and understanding about putting Chargoff's rule and Pauling's work together, and obviously even just understanding what incomplete data of Wilkins' work, or Wilkins and Franklin's work, incomplete data, how to put it all together. So I don't want to just completely diminish what they did. They still had to put everything together. There still took a lot of work to do that. But they were the ones who were given credit for it. And they gave in their final acceptance, kind of when it was kind of finally proven, they gave Rosalind Franklin credit just barely. They gave her the tiny bit of the credit. Kind of like at the end of a movie when they do the credits, she was like on the last line instead of being a major character because she clearly, all of her data helped. She was kind of on that last line of the credits. So here's the story of the scientists that kind of played their role into understand that structure of DNA, which opened the field up. Once we knew the structure of DNA, we could figure out how DNA replicated. We could figure out how the DNA cells replicate, and more importantly, how DNA can be used to make proteins, because their model that they created did all of that stuff. All right. All right, that's going to be it for now. That's kind of a long video here, but it's kind of a long story. The story literally lasted like 3 decades. So sorry, it took like 20 minutes. All right. Otherwise, that's going to be it. Thank you very much for listening. Good luck, and may the science be with you. See you.